Good evening, ladies and gentlemen. My name is Colin Lynch, and I'm a member of Sunnybrook's Board of Directors. It's my pleasure to welcome you to tonight's speaker series. As our last public lecture for 2021, I want to sincerely thank you for making these virtual events a great success. Since we made the switch to our virtual platform just over a year ago, we've been able to continue connecting with you, our patients, community members, friends, and family, to deliver relevant and interesting health information. As we continue to plan for the speaker series in the new year, please consider joining our online mailing list to get updates about upcoming sessions. You can also refer back to this webpage to get updates about the speaker series topics for 2022. It's quite apt that tonight's lecture is on the science of vaccines as we approach the anniversary of COVID-19 vaccine administration in Canada. We as a society have seen just how mighty the contents of these small vials can be, whether it is getting vaccinated against COVID-19 or getting your annual flu shot. Tonight, our leading Sunnybrook experts will be providing you with an overview of the science of COVID-19 vaccines and what we need to know to inform vaccine development. I would like to now introduce you to tonight's moderator, Natasha Salt, Sunnybrook's Director of Infection Prevention and Control. Thank you for moderating tonight, Natasha. I'll pass the mic over to you.
Thank you so much, Colin, and good evening to everyone joining us virtually for this last speaker series before the new year. As Colin mentioned, my name is Natasha Salt, and I'm the Director of Infection Prevention and Control here at Sunnybrook, and I'm super pleased and happy to be able to moderate tonight's evening discussion. Tonight we have an excellent lineup of experts from Sunnybrook on a topic that's been highly requested in the past few months. Tonight, our lecture is titled The Science of Vaccines. Like Colin mentioned, we've seen how much of a difference that vaccines can make in global health. Whether that's the flu vaccine or COVID-19 vaccine, people like you may have questions about how vaccines are developed and why they are so important. My colleagues and I are hoping to present some really interesting information to you to understand the science of vaccines. We will begin this evening with a presentation from Dr. Rob Kozak, who will be presenting From Boosters to B Cells, an overview of the science of COVID-19 vaccines. This will be followed by Dr. Samira Mubaraka, who will be presenting Viral Zoonotic Spillover, what we need to know to inform vaccine development. We have set aside some time at the end of the evening for you to ask our panel of speakers some questions. Thank you to everyone who's already submitted a question online before this event. Please feel free, however, to send your questions while presentations are happening through the website. We will do our very best to get through all of the questions in the time that we have available. So without delay, I would like to begin our event for tonight. And I'd like to introduce Dr. Rob Kozak. So Rob is a clinical microbiologist and Sunnybrook Research Institute scientist. He received his PhD in microbiology and immunology from McGill University in 2010. He has studied a number of different viruses like Ebola, Crimean Congo hemorrhagic fever virus, Zika, and now SARS-CoV-2. In the early days of the COVID-19 pandemic, Dr. Rob Kozak, in addition to Dr. Mubaraka, isolated the SARS-CoV-2 virus in a level 3 containment facility. It is now the principal source of SARS-CoV-2 to most academic CL3 laboratories across the country. We are so proud of them here, and I'm sure you will enjoy all the wealth of information they have to offer. Thank you so much for being here tonight, Rob, and over to you.
Thank you so much, Natasha. My pleasure. So welcome, everyone. I'm as well as the kind introduction. It's wonderful to talk to people about a subject area that I've been doing research on for a number of years. So I'm always happy to ramble about vaccines. I think that I think we'll all agree that it has been a long road since we had our first case here in in Canada back in January of 2020. I think this is one of the moments where we all kind of remember where we were when we saw this on the news. For me, and I'm sure for a lot of other people too, though, we also remember where we were that moment when Health Canada approved the first COVID-19 vaccine in December. So less than 9 months after we had our first case, we already had a vaccine ready to go into arms. And so when I was putting this together, I went to a lot of my friends and family and asked them, if you wanted to hear a talk about trying to understand these, what would you like to know? What are some of the things that are the questions that you have about this? And there were lots of questions about the science. I'm going to try and address as many as I can through this presentation. But one of the themes that came up a lot was, this idea that everything seemed very quick. And as I sort of dived into the literature and reviewed a lot and read a lot of things that are out there, and then based on my own experience, you actually come to realize there's quite a bit more of a backstory to this vaccine development, and I think it makes it even more interesting. So let's take a step back here. We're all going to go back to undergrad, or maybe if you did science undergrad like me, you certainly had to take a basic immunology course, and one of the main reasons or one of the founding principles of immunology is to understand how we can make a good vaccine. So I think we all remember, again, from maybe even from high school biology, something called the central dogma, where this is one of the cornerstones of science, where we know that DNA or RNA is made from DNA, and then RNA gets converted into a protein. And this is going on in your cells all the time. So this is the kind of one of the fundamental principles of biology, but it also really applies to vaccines, because ultimately the main goal of a vaccine is just to be able to show an antigen to your immune system. And if you show the antigen to the immune system in the right way, and it's the right antigen, then your body will develop B cells and T cells that are basically trained to then become activated when it encounters the pathogen in real life. And these different cells play different roles. The main role of B cells are to produce antibodies, and antibodies' main job is really to neutralize the virus, so bind to it and prevent it from getting into cells. And then T cells, their job is not only to be supportive to help the B cells in this process, but they also provide a crucial role by actually eliminating any cells in your body that are infected with the virus. So B cells their job is to slow things down, and T cells basically do the cleanup after. And when they work well together, you're able to be protected from severe disease, or sometimes you don't even notice that you actually have any symptoms. Now, the critical part of these vaccines is obviously vaccines only work well if you can show the antigen to the immune system prior to actually encountering the pathogen. So in the case of SARS-CoV-2, which is the virus that causes COVID-19, we need a good antigen that we could show to the immune system before you actually encounter the protein. So I'm sure by now everybody has heard of SARS-CoV-2. I'd like to just show this, because this is what SARS-CoV-2 actually looks like right here. I don't know if anyone can see my cursor here, but so this is the virus itself. This is an electron micrograph of the virus coming out of a cell. So that's what it looks like in real life. I find it a little bit easier, though, to show this kind of cartoon picture right here, which is a representation of the virion, and has all the different proteins, including what you can see here, these little orange guys, which are these are the spike proteins. So these are the component of the virus that it uses to attach onto the cells and get in and do all its horrible business. Now, remember what I said was, if you want that, all you have to do to have a good vaccine is show a good antigen to your immune system. Now, at the beginning, we were lucky that a number of groups actually sequenced the virus early in January, and the first genome was actually available on January 9th, 2020. So we knew what the sequence of the virus, and that's really handy if you're trying to design vaccines that express certain antigens. And early on, people realized, well, the spike protein actually might be a good antigen. But this wasn't just a lucky guess, because as you see here, there's a number, there's the envelope, there's the nucleocapsid, there's the spike. So there's a number of different proteins in the virus. So it's not like we just threw darts at a dartboard and said, well, I hope that this one works. This is actually something where we had learned quite a bit from research that had been done with the first SARS, so SARS-CoV-1 back in 2003, 2004. So this is a pretty complicated chart here. So let me just draw your attention to these two areas that are highlighted in the red box. So what this experiment was doing is they took hamsters and they vaccinated hamsters with a viral vector that was expressing the different proteins of SARS-1. And so what they said is if you use inactivated SARS, you get protected. It sees all the antigens, the hamsters are protected. But the only one where you saw protection, where you saw no virus in the lungs, was if you use the viral vector that had the S protein. So this really showed us that going forward, that the spike protein would probably be a really good antigen to use. So we applied that same logic when we started making vaccines against SARS-CoV-2. So in order to do something like that, it's actually we're quite lucky with the fact that we have so much types of genetic engineering and ways to synthetically make vaccines. So in the old days, you would have to take a virus and potentially you'd have to treat it with formalin, or you'd have to find out some way of inactivating it, and then you'd use that to vaccinate people, which is that was the rationale for the first polio vaccine, actually. But now we have the sequence of the virus. So all we need to do is find out the sequence for the key antigen, which, as I said, is the spike gene, the S gene. And if we just take that particular segment of the virus, if we make a synthetic version of that gene and put it into some sort of vector, we'll be able to deliver it to the cells. So again, let's go back to that central dogma where I said, if you want to show a protein to the immune system, you have three choices. You can give them DNA, and the DNA gets made into RNA, and the RNA gets made into protein, or you can give it RNA, and the RNA gets made into protein, or you can just skip the two middlemen and put protein back in. So the rationale behind the mRNA vaccines is, again, we deliver the mRNA for the spike protein into the cell. We do it through a lipid coat, which you can see right here. The lipid coat is basically a way for the mRNA to fuse with your cell. The mRNA gets in. Once it's in there, the cell says, oh, this is mRNA, I got to convert it to a protein, turns it into a protein, shows it to the immune system, and you generate your immune response. So this all seems, I guess, pretty simple. And again, it really did seem like we really kind of whipped through things, and we had a vaccine within 9 months. But there's a really, really good review that I would recommend to people who want to know more about this in Nature, where they actually highlight how long it actually took us to get to the point where we would have the technology for mRNA vaccines. So even though that it was as far back as the 60s where mRNA was actually discovered, the first idea for an mRNA vaccine was actually for influenza, and this was, people started doing experiments with this in animals in the mid 90s. And then from there was a lot of problems. There was a lot of optimization. What they found is they had to actually do modifications to the base pairs, the sequence of the mRNA in order to get it to be immunogenic. And then, of course, there was work being done by other groups that made this lipid nanoparticle, which was so critical for the delivery. So when people say, oh, we designed these vaccines, only took 9 months, it's like, well, not really. I mean, all the technology and everything that got us there, took the better part of 40 years and was, built upon incremental success from a number of different groups. But we were fortunate that we had all this available, and people were able to take that and some of the other knowledge that from SARS-1 and design these vaccines. We were also lucky that one of the things that you want to do for evaluating a vaccine is you want to be able to test it in animals to see if it's protective, because animals are situations where we can actually deliver the virus to them. In this case, give them an intranasal challenge, and then we can monitor them from symptoms, for viral shedding, for signs of disease. And if we see that the vaccine reduces or eliminates that, then we can say it's protective. And again, this was something where we built upon the knowledge that others had made available from studying SARS and MERS, which are other coronaviruses. So we knew that there were different animal models, including mice, ferrets, and non-human primates that would work. And then we later discovered Syrian hamsters as well. And so we were able to use these in order to, evaluate the immunogenicity and the protective efficacy of vaccines. The other revolutionary trick, I think, that was allowed us to get here was that normally in the vaccine process, you have to have the three-phase trials sequentially. So you start with a phase 1, which is relatively small, usually dozens of people, less than 100 often, and you will evaluate the safety. So does it cause an adverse reaction? You'll determine the dosage, how much vaccine I need to actually put into an arm. And if you get good data and everything looks promising, you then put together all your findings, you beg people for more funding, and you move into a phase 2. And a phase 2 is usually hundreds of people, so it's a little bit more expanded in size. And the goal here is to look at immunogenicity. So when I put that vaccine into someone's arm, do they generate antibodies? Do they generate protective T cells? And I also want to look at different groups. So does it provide a good immune response in children? Does it provide a good immune response in the elderly? So this was the next phase. And if you see promising data there and everything's looking good, again, you write up your findings, you go and you beg for more money, and you get into a phase 3, which is often involving thousands, if not tens of thousands of individuals, where you vaccinate them and you compare them to placebo to see if you actually see efficacy. So does the vaccine protect against any number of outcomes? It can be symptoms. Does it protect against hospitalization? Does it protect against death? Or do you look at all of them? So in order to shorten this process, what a lot of companies and groups did was, rather than doing them one right after the other, they lumped them together and did combined experiments, or sorry, combined trials. So you do a phase 1 and a phase 2 together. All you need is more people that are willing to do it. And it's all about the number in order to achieve the statistical powering. And this has allowed us to sort of skip ahead out of a lot of potential spots that might slow us down. And of course, it was obviously helped that a lot of the companies were well subsidized by government, so funding wasn't an issue. So basically what we have now is that all of our vaccines are based on again, this fundamental idea that I said of delivering the S antigen to your immune system. So right now there's at least 5 that are licensed. I believe only 3 of them are actually, or 4 of them are actually approved in Canada. They all deliver the S antigen. There's a little bit of variation in some of their doses, but we were also, one of the other things that we're quite lucky about is the antigen, the S antigen of SARS-CoV-2 is actually really good at inducing both a T cell and a B cell response. So there's epitopes, which are portions of the protein that are recognized by B cells and by T cells. And in some viruses, you don't actually have this. You don't actually have a good antigen that has both of these. And both B cells and T cells are so critical for protection. So common question from one of my families, so many different vaccines. I'm sure everyone knows WTF stands for, which of them is your favorite? And I would say that I early on and continue to this day sort of say that, I don't believe that there's a huge difference between the vaccines that we're using here in Canada. They all provide similar levels of protection. There has been some data that has come out recently from a study in the US that's showing that maybe Moderna and Pfizer have a little bit higher levels of antibodies, both neutralizing and other antibodies. So neutralizing antibodies, their job is to protect the virus from getting in. So they might induce a little bit higher, but the Johnson & Johnson one still does provide efficacy. The other thing that this study is actually interesting, because even though the Johnson & Johnson may have a little bit lower efficacy at hospitalization, it's prompting people to say, well, what if we give a second Johnson & Johnson shot? Or what if we pair it with some of the other vaccines? Is that going to lead to better protection? And I think most importantly, the take-home message is that, whether something is 93% efficacious or 88% efficacious, if we get everybody vaccinated, then it will matter a lot less. Okay, so one of the things that I'm sure everybody has been seeing quite a bit of in the news recently is this focus on boosters. So the rationale for this is that there have been a number of studies that have said that we don't fully understand what are called the correlates of protection. So we're not sure exactly what things we can measure in your immune response that we know will mean that you're protected. But one of the things that we think it might be is these neutralizing antibodies. So if you have high levels of neutralizing antibodies, we know that you're probably going to be protected better against not just against severe disease, but you're also less likely to transmit it to others. And there's been a number of groups that have actually looked at this and said, over time, what was noticed was that the quantity of your neutralizing antibodies goes down. So what is seen is that if your vaccine is about 95% efficacious, so that's comparable to what the Moderna or the Pfizer initially were, by about 250 days after you've got your vaccine, the antibody levels have gone down. And so likely you probably, the vaccine is only likely about 70% efficacious. And so this was actually done by this group right here. This is the figure that I'm referencing. And they did this entirely by modeling based on clinical trial data. But then a number of groups in Israel and some groups in the US found similar things where people's level of neutralizing antibodies actually decreased. And the group in Israel showed that, so if we take these individuals, and they focused on individuals greater than 60 because they were more at risk for severe disease, if you gave them a third dose, a booster, that they had a reduction in their disease severity by a factor of 5 on day 6 and a factor of 11 on day 12, and overall their risk of severe disease was reduced almost by a factor of 20. So they are less likely to get infected, and they are less likely to have severe disease. And most likely during this time, when after you get the booster, you're less likely to transmit it to somebody else. So this is why there's a lot of push now on these boosters, because it's probably a good thing to do during the winter influenza virus season. We've got lots of people sort of indoors, lots of potential for transmission. So if we keep those neutralizing antibody titers up, not only are we going to hopefully keep people out of hospital, but maybe likely limit transmission as well. Also, what has been shown is that if you give additional boosters to individuals either who have been vaccinated or individuals who have actually caught COVID and recovered, what you actually do is you generate really good memory B cells. And so these memory B cells are basically, these are the cells that hang around so that 1 year or 2 years or 10 years later, if you ever encounter a pathogen, these B cells remember it, and they will generate antibodies that will be specific for that particular virus. This is why, for example, you have protection against hepatitis B decades after you received your hepatitis B vaccine. So a study done by a group in Europe showed that with this vaccination, not only do you increase the number of memory B cells that have specific antibodies for the receptor binding domain, so the part on the spike that's designed to attach onto your cell, you get really good antibodies that bind and prevent that, but also you get lots of diversity. So the antibodies that you generate are going to be able to likely bind to other variants. So this gives us a larger and broader depth of potentially of protection with these boosters. And you can imagine a situation down the line where if COVID is going to be with us forever, and there's in all likelihood it's going to become an endemic virus that we're stuck with, you can imagine us potentially pairing our boosters for COVID-19 every year with our flu shots as a way of doubly protecting everybody. So I think too that the other thing, we can't talk about boosters without talking also about kind of the good news that we've seen recently, where we've seen that Health Canada has just recently approved the Pfizer vaccine for all the way down to age 5, and I'm sure the Moderna vaccine is not too far behind it. And a lot of things that a common thing that I encountered with a lot of my friends and family was, people said, COVID doesn't seem to be so serious in children, doesn't seem to be, as bad. And I think this is something that, that this is if we actually look at the data, we see that, it's not quite so cut and dry. So there's no question that in older people, that COVID is much more serious. But we look at the data that we have here in Canada, and over 370,000 kids have got COVID, and other countries have shown that maybe up to 20% of them may have the symptoms of long COVID. So even after they recover, they're never, they're not fully at 100% 6 months later. We've also seen almost 2,000 hospitalizations, and regrettably, we've seen 19 deaths. So this isn't really something that we should dismiss. We shouldn't think of it as something that is, has no effect on children. And that's why I think that it's such great news that the Pfizer vaccine has been shown to be efficacious. It's 91% effective, and that it is safe for use in children, because I think not only will it help protect them, but also there's a way it can be used to prevent the transmission to other members of the household. And I'll just also highlight, this is actually from something for a common thing is, if it's not that serious, why bother vaccinating against it? When we compare COVID-19 in children to other diseases where we have developed vaccines against, we've developed vaccines for, diseases which are far less lethal than COVID, because there's a definite improvement on cost or on quality of life. We reduce morbidity. It's good for the public health good. So I think there's absolutely good reason to do the same for COVID. Okay, so I'll just kind of end this now by hoping that what you've taken from this is sort of an introduction to the science. I could spend 2 hours talking about the science, but I definitely will not. I think what this story has really shown me is that the path to these vaccines was not sort of done overnight. This was built on incremental steps, and truly the people that did it were standing on the shoulders of giants. It's also kind of shown us that we can develop vaccines in record time. So I think the question shouldn't be why is this vaccine why did it only take a year to get licensed when other vaccines take 4 or 5? It's why are we letting other vaccines that could be saving lives take 4 or 5 years to be developed when we can do it faster? And I think there's still lots more to discover. I mean, this is, as a scientist, it opens up all sorts of questions. Can we make a universal flu vaccine? What about one that we could use to protect against all coronaviruses? And how can we be ready for, the next thing that comes, the disease X, the one that we haven't prepared for, because this will not be the last novel virus that we encounter. And I think it's important to remember that it's the research of today that is critical for preparing for the pandemics of tomorrow. I think the mRNA vaccine story showed us that. And I'll just leave you with this last slide, just to encourage all of you to become a citizen scientist. I always tell my friends and family, and I encourage everybody here, follow the scientific and medical literature, read the papers, look up the journals. They're not hard to follow. You can, they have great summaries. Ignore the trolls on Twitter. When you're reading it, ask yourself, what does this data really say? Challenge yourself, learn more about it. It's never a bad idea. Things will change, guidelines will change. We just have to be, we have to be patient. It's always best to talk to experts and use common sense. None of the things that we've done are going to actually on their own get us out of this. We can't boost our way out of it or mask or test. We have to use all of them, and we have to use common sense. And just remember that in the end, this too shall pass. So I'll just explain these. My niece, I've worked in, as Natasha said, I've worked with a number of high consequence pathogens. And the weird thing is you can't take a picture of yourself in the suits when you're doing it, although you can find pictures of people on the internet. So I've had my niece, every time that I've worked on a different pathogen in a different lab, draw a picture to represent what Uncle Rob looks like. So this is me with Ebola and me with COVID-19. And with that, I think we're saving questions for the end. So I thank you all so much for your time.
Rob, thank you. Excellent presentation, really well done and super informative. I love the space suits at the end. Very nostalgic from period to period. I think that it's what I gained a lot from your presentation. I think it was fascinating. I love immunology. One of the things, that I keep getting asked as well over and over from friends, families, is what you touched on, which is the fact that everything seemed to evolve so quickly. Can we trust it? And, I've done a lot of reading as well, and research on the topic, and seen how much collaboration and support went between even pharmaceutical companies to try and get this moving, and collaborators, everybody really teamed up to against COVID is what I'm trying to say. I think, that made a big difference. And then there was, just the ability to keep going and forging ahead and working through every step. They didn't miss steps. They just were expedited and had the resources to be able to do so. And I think that that's what's really fascinating is, right, you're absolutely right. What else can we do when we know we can achieve so much in such a short time? I think that's a really great take home message here, is that we have the ability to do this and do this safely. And I think that's super important. With that, as Rob did indicate, we are going to be collecting questions and going through all the questions that were already submitted, plus those that you may come up with throughout listening to this presentation. So please keep sending them in. With that, I will introduce you to our next speaker, Dr. Samira Mubaraka. And Samira is an infectious disease physician and virologist and Sunnybrook Research Institute scientist as well. She has completed her MD at Dalhousie University and medicine training at McGill University. Since completing her research fellowship at Mount Sinai School of Medicine, she continues to study themes of viral transmission and spread. In the early days of the COVID-19 pandemic, Dr. Mubaraka, along with Dr. Kozak, isolated the SARS-CoV virus in a level 3 containment facility. It is now the principal source for SARS-CoV-2 to most academic CL3 laboratories across the country. And I think that's really important to remember. I mentioned it earlier, but I can't emphasize how fantastic that was when we achieved that here. So thank you for being here tonight, Samira. Over to you.
Thanks so much, Natasha. I'm just going to put this into slideshow mode. Hopefully you'll be able to see it. Might just take a second. There we go. All right, thanks so much, Natasha, and thank you so much, Rob. It's really great to be doing this with you. Almost 2 years since the first case arrived, and I have to say it was Rob who raised this whole possibility. I think it was over the holidays or right before the holidays that Rob, you sent that ProMed link around to everyone. So you were the first one, at least in our group, to sort of say, hey, we better keep an eye on this. And yeah, you were more than right. Unfortunately, in some ways, hey, I think it's good that we were somewhat prepared and had it on our radar, but the scale of it was something I don't think any of us were anticipating. And just before I start, Rob mentioned being a citizen scientist, and it made me think that folks can actually join Flu Watchers. If you just Google Flu Watchers, it's through the Public Health Agency of Canada. And part of the reason this triggered is because migratory waterfowl, so these flying geese, are actually reservoirs. They're the reservoir for influenza viruses. So if you're keen, I'm a participant. It probably takes less than 30 seconds a week to participate in Flu Watchers. So if you're keen on citizen science, please think about joining Flu Watchers. Okay, so we're going to take a walk on the wild side, and I know it might seem a little bit far-fetched to make links between wildlife or animal health and vaccine development, but I think as we discuss this, hopefully you'll be convinced by the end that really the two of them are very, very closely interconnected. Before we get into it, though, I do want to highlight who actually does all the real work. I mean, we worked so hard in a collaborative manner, as Natasha mentioned, but the essential researchers at the bench, at the bedside, at the console, at the command line are all these undergraduate students, these graduate students, techs, postdoctoral fellows who really have been so incredibly dedicated, and they're doing this all over the city, the province, the country, all over the world. So these are the people that are keeping the research going, and we owe so much to them. So thank you. So one of the things I want to highlight at the beginning, you always worry when you talk about wildlife as a potential source of zoonotic pathogens, that people will perceive wildlife and nature as a threat. And in fact, it's the complete opposite. Obviously, humans are the threat to nature. And a lot of our behaviors are what's driving viral spillover from an animal reservoir, its natural reservoir, its natural habitat, into other species like humans. So this is why I raise this up front, is that really it's not so much that nature is a threat to us. It's more about how we are driving changes in viral habitat leading to spillover events in humans. So a zoonotic pathogen is a pathogen that will spill over or start transmitting from an animal to either another species of animal or to humans. We are also animals. We should identify as mammals as well. And then there's reverse zoonoses, which this pandemic has clearly been associated with. So what that means is that the virus can go from humans into other animals. So these are really complex systems. It's more than just about movement between species. It's all about the impact on these species. Excuse me. And for example, the impact on humans has been very significant and underscored by a lot of inequity along the way. So one approach that we can often take to these kinds of complex problems is an approach that we call a One Health approach. So that includes looking at human health, but not in isolation, along with animal health and ecological health. Excuse me, I don't have COVID, just a dry mouth. And we have to look at all of these in the context of the biosphere we share, because the virus is actually moving between these. And it's not just about this sort of Venn diagram with 3 different populations and how they overlap. There's a lot of depth to it too. So biology is fundamental. You need to understand the biology of the pathogens themselves and the ecology of the species that harbor them, and obviously the immunology and the host response. But how the impact affects these different populations depends a lot on how we govern our laws, our policy, our economics. So that's really what One Health is taking an approach that includes all of these, but not necessarily for all solutions and problems. It's just making sure that you have more than one perspective when you're approaching problems like viral zoonoses. So this is really a very brief overview. We think that the reservoir for SARS coronavirus 2 is a horseshoe bat, potentially other species of bats as well. It clearly has spilled over into humans. I'm using our man Darwin here as an example. And then from humans, we know now that it's spilled over to deer in the United States. We know it has spilled over to mink. And we do know that there's been transmission back from mink to humans. So, and along the way, the virus has been changing as well. So there's a lot going on here. So let's start at how humans and animals could potentially be interfacing. What are some of those interactions that enable viral zoonoses to spill over? So some of the interactions are more altruistic. You can see here, it could be through animal rescue and care. Some of the interactions are fairly innocent and just driven by curiosity, but they can happen through an intermediate like a companion animal or livestock animal from wildlife. Other interactions are more nefarious, at least on the part of the humans. So you can have urbanization, you can have massive industrialization, and you can have legal and illegal wildlife trade. So all of these interfaces can enable zoonotic spillover. And urbanization and industrialization merit highlighting for a couple of reasons. What's been happening worldwide has been massive industrialization that creates what we call these edge effects. And this picture really sort of exemplifies this. You have very large areas of wildlife habitat really abutting areas of very, very dense human population. So you can see that should any of these waterfowl or birds harbor an avian influenza virus, it's not that far to go to find a human host when you look at what's in the background here. So this is what we call edge effects. And the larger the edge effect, the higher the risk of spillover. These are a couple of examples, but probably the most significant example of how we interact with animals is less so on the wildlife side of things, more so on the livestock and agricultural side of things. So from an agricultural perspective, some of it could be recreational. You know, people do goat yoga, people go to petting zoos. We interact with livestock in many ways. But probably one of the most significant ways we interact with livestock is really through our food chain. And it could also be through our occupation. So if you look up here, for example, this is a highly industrialized and intensified farming operation. So for example, in Canada, swine farming a couple decades ago consisted of more but smaller operations. Now, this particular picture is not in Canada, but now you see much larger operations with very, very high concentrations of animals in one place, which makes the risk of spread quite high, depending on whether or not disease control is possible. And I have to admit, we've done some work on swine farms, and there's more biosecurity for us to walk onto a swine farm or go onto a swine farm than there is to come into the hospital. We have to change our boots twice. And if you do have a vehicle that goes from one farm to the next, you actually have to get your undercarriage cleaned before going to the next farm, so that you aren't bringing agriculturally important pathogens from farm to farm. So biosecurity in farming is incredibly important. And also, the other setting to really highlight here is live or wet markets, where you might have animals coming from different geographic regions into one particular site. And you may have also multiple species all interacting in the same site, and the public is there as well. So you can imagine that live or wet markets are a high risk setting, in addition to the other ones that I've already mentioned. So why does this matter? I like this figure because it actually puts the virus at the center of the figure, as opposed to the human, because really it's all about the virus. If you follow the virus, you'll see it'll uncover a wealth of knowledge around how transmission happens. So this is a little bit older, but again, it's a nice sort of bird's eye view. We can see that, there are a number of different viruses. For example, H5N1, H7N9, those are both highly pathogenic avian influenza viruses. Then there are what we call the high consequence coronaviruses, like MERS and SARS. And then there are the Nipah viruses, so Hendra virus and Nipah virus, which the WHO has highlighted as pathogens of significant concern. And really, the virus finally gets to the human through at least one, if not two, intermediates. The first one's usually a reservoir. So we know that bats are fantastic reservoirs for many viruses. And I'll briefly mention why in a second. And then there could, or perhaps not, or it may be unknown, be an intermediate host. So for example, with MERS, it's pretty clear that humans are not necessarily getting infected directly from bats. They're getting infected from their interactions with camels and sometimes with each other. Similarly for SARS back in 2003, because of the genomic data that's now available, there's very good evidence that the intermediate host was a palm civet or a civet cat. And the data shows that it probably spilled over from a bat to a civet cat at a farm, and then that civet cat came into a live wet market, and that's probably where the spillover happened into a human. And likewise, for some of the Nipah viruses, there are other mammals that are important. Pigs for Nipah, horses for Hendra in Australia. And obviously for avian influenza viruses, there are avian species, mainly poultry. So how these animals, the reservoir animals and intermediate animals, are affected or impacted can also impact on how viruses spill over. So one example really is around for Nipah virus and Hendra virus. So changes in foraging, so that's when bats go out and start to feed, can actually be caused by deforestation, plantations of things like orchards for frugivorous bats or bats that feed on fruit. And that brings them into closer proximity to either horses or humans than normally would happen. These are flying foxes here in Australia, and although they generally roost in trees, they're usually much more spread out. These ones are roosting in these big clumps, somewhat paradoxically, because they're suffering from heat stress. So they go into this big clump for, again, you think that wouldn't be the solution, but this is a behavior that's being observed, which can definitely change any kind of viral dynamic that these flying foxes may have. And people are finding flying fox die-offs at the base of these trees because they're dying of heat stress and then are just falling down to the base of the tree. So anytime you anytime you affect the behavior and the wellness of a bat or a reservoir host, there's a risk that you're going to change the viral ecology. And so I have probably a more concrete example here. This is a site in Belize where a number of bat biologists, and we've been down there as well, go to look at bats and understand bat biology and virology. And before this was all forest cover. A couple of years later on return, you can see that there's been a clear cut. This was done to raise cattle. So a couple of consequences here. The tree roosting bats are really, really severely affected here because their habitat is just gone. So biodiversity gets lost. But then some bats, like this Desmodus rotundus, it's a vampire bat, are absolutely thrilled because they don't depend on trees for roosting. They like to roost in cave ruins. And in this case, this is a looter's tunnel in a Mayan ruin. So their habitat's not affected, but now they've got fantastic access to food. They feed, they're hematophagous. That means they lap up blood. So they, now they've got these, an almost infinite source of food. And because of this, there've been a couple of outbreaks of bovine rabies because Desmodus rotundus harbors rabies. And with their populations flourishing under these conditions that were really, there's a good chance they were secondary to that clear cut, are able to spread more virus. So bringing viruses that tend to live in reservoirs in fairly diverse populations closer to domestic animals and humans increases the risk of spillover. So this is exactly, or what we think happened with SARS coronavirus 2, most likely spilled over from a bat clearly into a human. And this was the first case here at Sunnybrook that everyone on this panel was involved in. And so there's no question now that a spillover event happened. We don't know when it happened. Clearly there was human-to-human transmission by late 2019. But again, we have no idea when the actual spillover event happened. As Rob has already highlighted, the virus was isolated at the University of Toronto, and you can see it budding here from these cells. And this is essentially the folks, some of the folks in the biocontainment lab that were working on this in collaboration along with Rob as well. And very early on, even before that first case, what was incredibly helpful was that the Chinese government and public health officials released the genomes of the viruses that came from those cases of acute pneumonia. And you can tell how old this is because it still refers to nCoV, novel coronaviruses. That was even before we called it COVID-19. And the reason this was really important was multiple. Number one, at least we knew what we were dealing with. We knew that it was a SARS coronavirus. It was very closely related to SARS from 2003, but even more closely related to some other bat coronaviruses. So that sort of pointed to the origin. Number two, we had sequences, so we can now establish diagnostic tests because you have to know what the sequence is to develop those tests. And number three, you start to get a sense of what the antigenic sites for a vaccine could possibly be, as Rob has already highlighted prior to this. You had the entire genome to look at, and based on some of the information that was available from work from SARS, the first SARS, people had a sense. So this is all done through what we call next generation or whole genome sequencing. And just very briefly, what we do is we enrich the viral RNA. These are RNA viruses. Once you have the sequence, you know that you can develop these, what we call PCR primers, to enrich specifically for that, which increases your yield substantially, and sequence these little pieces that then you kind of stitch together to make the whole story, so that you have an end-to-end story about what is actually the whole genome sequence of the virus. And this has been incredibly important because we found a whole bunch of different virulence factors, many of which were not described before, at least in the first SARS coronavirus. And that's really helped us understand why this virus is more significant. Now, spilling back into animals is something that this virus did fairly early on. You may recall that there were outbreaks in mink in Europe. These were farmed mink. And then more recently in the US, I'm showing this study as an example, there was a spillover into white-tailed deer. And what we're finding is that, what the authors here found, was that the sequences from the virus that was detected in deer really reflects what was circulating in the population. There were a couple of clusters, so it's possible that there was deer-to-deer transmission. But the other reason that this is really important is because we need to follow what the spike sequence is doing while it's transmitting in deer in case it spills back into humans, and whether or not there's any risk of immune escape or vaccine escape from any viruses that have adapted more to deer as opposed to humans. So really critical piece of information there. Just to move very briefly away from SARS, and I'll wrap up quickly. I'm sort of lost track of time, but hopefully there are a couple of minutes left. I just want to underscore that we can't stop thinking about some of the other viruses that are circulating. And in fact, since May of 2021, signals for highly pathogenic avian influenza virus, we're calling it H5N-X. The reason for that is there are multiple neuraminidase subtypes that are co-circulating, which of course is incredibly concerning, to the point where in November we're seeing more alerts now. So OFFLU has issued a statement, and the FAO, the Food and Agricultural Organization of the United Nations, also issued an alert. Very concerning also has been spillover of avian influenza virus, highly pathogenic avian influenza virus, into mammals. Usually this virus should not infect mammals, especially in nature, and a couple of red foxes have died in the Netherlands from highly pathogenic avian influenza virus. So this is incredibly concerning, and in some places, for example in Ghana, they're seeing co-circulation of multiple highly pathogenic avian influenza viruses, and we're helping out just in small part with some of the work that's being done there. So at the end of the day, vaccine development really depends on us understanding what the wildlife reservoirs are, doing the surveillance in those animals so that we can anticipate what vaccines are needed, but also not to forget that we can also vaccinate animals, not just humans. So that's really something that places like Vito, which is in Saskatchewan, have really focused on developing both human as well as animal vaccines. And this can be done in parallel, in fact, and there's some really good examples. You know, rabies is a great example most people would be familiar with. Both animals and humans get vaccinated for rabies where indicated. And then there are a number of other viruses and bacteria as well where this could be important. And I'm just going to highlight one particular virus called Rift Valley fever, which is transmitted by mosquitoes and tends to infect ruminants. So these are goats and sheep, and can also cause significant illness. So they get abortions and fetal malformations. So during outbreaks, you might actually see what they call abortion storms in livestock, which can herald infections in humans. Humans can have mild illness, or they can get hemorrhagic illness and encephalitis. And a lot of Rift Valley fever activity has been associated with human activity again. So climate change, which of course increases mosquito abundance and feeding. Armed conflict compromises surveillance programs and livestock health programs. And then the animal trade. The animals get moved around, and that's another way to actually spread Rift Valley fever. So that's why it's so important, and why here people have highlighted the need for vaccinating both both humans and animals for Rift Valley. So just to briefly summarize, there's a significant role for pathogen surveillance. Understanding ecology is really critical, and taking a One Health approach is really essential. We really need to focus on preventing more viral spillover if we want to prevent the next pandemic. And one thing I really want to emphasize is how important our own, Natasha, you mentioned global health at the top. That's absolutely critical. We really can't be talking about vaccines without talking about vaccine equity. It's so incredibly unfair that, some of us are getting our third dose and some of our colleagues in the global south have not had their first. And this is something that people are striving to address through the pandemic treaty, but that will be too late. We need to address it now. So I will stop there, thanking many, many people and leaving you with this message. Over to you, Natasha.
Thank you so much, Dr. McGreca. This is an excellent presentation, and it just emphasizes what a delicate and complex ecosystem we live in. And I think now more than ever, we've all been focusing on all the public health measures that is required of humans, and it's really been consuming all of our attention. But this presentation really does show us how much of the entire system needs to be considered when we're looking at trying to prevent the transmission of diseases, and how we have to, advance ourselves in all these categories in order to kind of inform a better response in general globally. So thank you very much for that presentation. This does bring us to a conclusion for our formal presentations for this evening. And now what we really would like to get into is the questions that you have all submitted, either early on in the beginning when this was posted or during the presentation today. We have quite a good amount of questions, so apologies in advance if we're not able to get through all the questions. But certainly we're going to try and do our best to try and get there for you. So I'll get right into them without delaying any further, and I'll just bat them between the two of you two, and then feel free if either of you have anything to add to one another's questions, by all means. So the first question here today we have is, do you think that the COVID-19 vaccination will become part of the childhood boosters and shots? Maybe Rob, I can start with you, and then we'll go back and forth.
I kind of hope so. I can't definitively say. There's obviously at both the provincial and the national level, there are organizations that will decide based on a number of factors whether or not the vaccine should be part of the routine immunization schedule for children. I hope it do. I have friends who have children who have comorbidities such as asthma, and I want them to I want those kids to be protected, and so I can imagine that since COVID will be around with us probably for a good long while, that it just makes sense to do that. As I showed in my presentation, like the last thing we want is kids hospitalized with COVID. So if we can vaccinate them and protect them against that, I think it just makes sense.
Absolutely. I think, Rob, to that, it is very important that children are vaccinated, but in the same breath, I think our adult population as well, you could have that same conversation, and just similar to our flu vaccination, I think you'll agree, if it is something that is available to us, I think it's there for good reason, and that a lot of work has gone into it being available to us and being offered. And I think it's important that it is, administered to all those age categories.
Absolutely, absolutely.
Okay, Sam, maybe this one I can head in your direction. So when it comes to the development of vaccines, do studies look at the short-term and/or long-term effects on the female reproductive system?
Yeah, so I think it depends on the vaccine and also the study, obviously. But for example, looking at vaccines for SARS coronavirus 2, the studies have looked at rates of conception for people who received either placebo or or vaccine and found no difference. So that's just an example of one of the things that people can look for. Because I know that fertility has come up, and I think that the vaccine manufacturers want to address it immediately as soon as possible, because obviously the implications would be significant. Fortunately, from a biological perspective, there's really no compelling reason to think that it would affect fertility. You know, these, as Dr. Kozak already explained, you know, the mRNA gets degraded pretty quickly. It, you know, you makes the protein, you raise the antibodies. So really in that, from that perspective, you know, there was limited cause for concern, and I think the clinical trials so far have shown that that there's no clear cause for concern.
I'd like to add one thing to Dr. Marca's comment. In the Pfizer trial, I believe there were 2 or 3 women who actually got pregnant during the trial. So I think that's strong evidence suggesting that it does not have an effect on fertility.
Thank you for that, both of you. Okay, on to the next question. Can blood work indicate the level of antibodies someone has after receiving the vaccine? And that goes for either COVID-19 or flu vaccination. Maybe Rob, I can give that one to you.
Sure. So we do have we have a number of different assays that we can look at the amount of antibodies that are present following vaccination, and so these are actually used in clinical trials. Remember, I said the phase 2 clinical trial, they measure immunogenicity. So we have assays that say here's how many. It's called they look at the titer, and we can do that for COVID. They actually use that for influenza on a regular basis to see how well a particular flu isolate is interacting, like how well your antibody titer is against a particular flu isolate in order to potentially modify the vaccine. So the problem, though, or the limitation of it is, and I don't want everybody to now rush out and go check your antibody titers, is that especially in the case of COVID, we don't fully know what level of antibody is actually fully protective. We're starting to get an idea, but we don't know exactly. And there may be specific types of antibodies that aren't measured through our conventional assays, where you actually have to take the antibodies and put them together with viruses in a level 3 and see how well the viruses are prevented from infecting. That's how we look at neutralizing antibodies. So that's not something that we can capture through our conventional testing.
Okay, that's great. So basically the short answer is that yes, there is a way to look at the antibody levels. And I think that's historically done, is it not? Looking at antibody levels for various vaccines in general. It's not a new thing, right? So it's common.
Sorry, yeah, I guess I'll just elaborate a little on that. So for some viruses, yeah, we know for hepatitis B, we know exactly how much antibodies are required to be considered protected. We know the same thing for for mumps, for measles, but that information obviously came through decades of research and trials. But yeah, the idea of measuring antibodies to look for protective efficacy is something done routinely in a lot of clinical labs all over the world.
Great, thank you for that. Okay, Sam, I think this one is right up your alley. So what happens during human to animal transmission? Is it something that I should be concerned about with my household pets?
Yeah, so it's certainly possible, and it sounds like cats are potentially more susceptible, though I think dogs have also been shown positive as well. And if you want a really good reference, someone by the name of Scott Weese, W-E-E-S-E, has a great blog called Worms and Germs. He's basically an infectious diseases veterinarian at the University of Guelph at the Ontario Veterinary College. And he has some pretty specific advice around self-isolation and isolating of animals if need be. So it's certainly possible that there is transmission. Now, fortunately, we're not seeing, profound illness in companion animals, which is great. But I did hear that maybe some larger cats have succumbed to SARS coronavirus too. So, and by larger cats, I don't mean companion animals. I mean larger zoo animals. So we still need to take measures to prevent spillover, but it's certainly possible. One thing I learned during this pandemic is that there's a coronavirus that can infect cats, and it has an enteric form, but if it acquires that same, what we call the polybasic cleavage site, is that we see, or very similar to what we see with SARS coronavirus 2, it can actually be fatal in cats. So again, this is a completely different type of coronavirus, but just to kind of underscore that so many animals have their own coronaviruses that can cause significant disease. So in that case, it's called feline infectious peritonitis virus. But yeah, so I think that preventing, if you can prevent your companion animal from getting sick, then obviously that's best. But so far, illness has been pretty limited.
So I guess physical distancing to dogs and cats all apply as well. Try and minimize contact. I haven't seen any dogs or cats with masks yet, but maybe that'll be the thing of the future. Okay, on to the next question. Rob, maybe I'll start with you on this question. And there's actually another question that came in online that I think kind of goes well with this question. So maybe after the first part, I'll continue on with you, Rob, and you can answer the second. I think they go well together. So when we hear things like herd immunity, how much of it really depends on vaccines?
Well, so first off, I mean, it often depends on the pathogen. There's a historical study that was done on an island in the North Atlantic amongst a population that had where measles broke out, and they found that after a certain generation died and there were new people and measles was reintroduced, all those that they lost their herd immunity because everybody who had immunity was no longer alive or on the island. But for other pathogens, immunity tends to wane. I think in the so like something like measles, while conferring lifelong immunity, something like influenza, for example, we know we don't have lifelong immunity following exposure. When we think of it in the context of COVID-19, there's a lot of debate between natural or I won't even say natural immunity that's acquired through infection and immunity that's acquired through vaccination. We do know that immunity that's acquired through infection doesn't last as long, is generally less protective against variants, and by no means confers lifelong immunity. The vaccines, we're still learning how long they will actually protect for. But I think that there's data showing that at least you've got at least 6 to 8 months of really good protective efficacy. So if you want, the goal of herd immunity is to have your entire population immune. And the trouble is we don't know how many people or what percentage of the population that is. We know for something like measles, you have to have at least 85% of people immune or the virus can spread. We don't know what that is yet for COVID. It may be something incredible that we may need 100%. We may not be able to achieve that. All that being said, I think that if you're looking for what is the better way to develop immunity, most people who survive any infection will have some level of immunity to it for a set period of time. But if you can get immune easier without getting sick, without ending up in hospital, I think that is the better way to go.
Mm-hmm. Exactly. Thanks, Rob. I think, second to part of that question, which I think you've also touched on, was a bit around, the question was, is there such a thing as natural immunity? So that would be obviously the infection. And what does this mean for people who cannot get vaccine because of previous medical conditions? Do you want to add to the conversation there?
So this is one of the reasons we push for herd immunity is we realize that in any population, there are going to be individuals who either the vaccine just won't work because of their immune system, or they won't be able to take it for any number of reasons. But and that's for example, the rationale for measles. If you have 85% of people protected, the ones that can't get it should still be protected because the virus has nowhere to spread because you've got the 85% covered. So that's what we're pushing for with herd immunity in order to protect those that can't necessarily benefit from it. I'm confident, though, that as we continue to improve our COVID vaccines, and people are already doing research onto the next generation of them, there may be new versions of them, new types of vaccines that may work better in people who are unable to tolerate the current COVID vaccines. Although that's a very small amount of people from what we know right now.
Thanks, Rob. So I think what I'm hearing is really get vaccinated, whether or not you've had COVID, get vaccinated. Unless you have some sort of contraindication. We're learning a lot about this virus, and more information will come in the future, but the bottom line is I think getting towards herd immunity is definitely a mix of having the infection and or being vaccinated, but preferably both. I mean, not both. Not necessarily wanting to be infected with COVID, but definitely at least having the vaccine.
And if you get infected, as I showed on the one slide, you get vaccinated after, and your immune response is going to be even better. So there's no reason not to get it.
Exactly. Great, thank you. So Sam, I'm going to shift this one to you. So is there a possibility of an influenza virus causing a pandemic? And if the genomes mutate or shift, can a more pathogenic strain emerge?
Yes, and actually, I mean, influenza is usually the one we worry about, right? We really worry about the next influenza pandemic. The last one was in 2009. Very fortunately, it was mild as far as pandemics go. This was the first coronavirus pandemic, at least that in recorded history that we know of. But yes, I think probably that was addressed a little bit in those slides. We're seeing more highly pathogenic avian influenza virus activity ramping up in the latter half of 2021, which is concerning enough that people are issuing alerts. And you're right. I mean, you did or the person who asked the question asked about changes in the genome. And there are a couple of ways that can happen. You can get like a gentle drift, so single mutations, and that's usually what we see with seasonal influenza viruses. But when we see big, what we call antigenic shifts, that's when entire chunks of the genome, it's a segmented genome, reassort. And that usually happens in an animal reservoir. It could be a swine, or it could be some other what we call mixing vessels, because avian influenza viruses tend to infect avian species. Mammalian influenza viruses tend to infect mammals, but they can reassort, and you can have an avian virus that's capable of infecting a mammal, i.e. us. And if that particular virus is one that we've never seen before, so that's one of the conditions for a pandemic to happen, is that we have to be susceptible. It won't cause a pandemic if it's a virus that looks a little bit like one that we've seen before. So we might have antibodies to, even if they're not 100% protective, they might be partially protective. So for example, I've probably never, well, almost certainly have never been infected by an H5 influenza virus. So if I were exposed, that there would be really high risk. The two other conditions for a pandemic is that it has to cause disease and it has to transmit person to person. So the virus would have to mutate in such a way that person-to-person transmission is enabled. And we've already seen some H7 viruses with those traits. And that, again, is why surveillance is important, because now that we know what those transmission determinants are, we have to keep sequencing the viruses that we find so that we can figure out what the risk is.
Thanks, Sam. I think, this question was an excellent one. And I do, all along, we've all been preparing for an influenza pandemic. And historically, that's exactly what we saw, was these shifts where pandemics would arise, and they were always influenza. So this one caught us a bit off guard in terms of the virus. Fortunately, the same sorts of principles apply with respiratory viruses. So I think we were able to easily shift and adapt, but just interesting how history has evolved. And we went from SARS-CoV-1 back in, 2003, that gave us a little bit of a scare. And then all of a sudden, this big pandemic now. So interesting how things change and how the unexpected can arise anytime with viruses. They don't give us any warning. So maybe, Sam, I'll keep with you on this next question as well. Can transmission of a virus change over time? We've been dealing with COVID for almost 2 years now. Do you think the biology of the virus will continue to change?
Yeah, so unfortunately, I think the variants of concern have shown us how it's done, right? We're seeing what we call the R naught or the reproduction number increase with the variants of concern. So there's certainly a risk that there'll be enhanced transmission, and we're starting to understand what parts of the genome are contributing to that. I briefly alluded to what we call a polybasic cleavage site, which the virus really seems to depend on for good replication, which can also lead to better shedding and transmission. So unfortunately, yes, the virus has been changing. Now, more recently, and I'm knocking on wood, there'll probably be a preprint tomorrow that will say the exact opposite of what I'm about to say. But since Delta emerged, it sounds like at least the accumulation of changes in S slowing down a little bit. Again, I'm always prepared to be wrong, and it could easily speed up again. We just can't predict what this virus is doing. But we can also try and get things to reverse order, and if not reverse, at least slow down. And again, it comes back to vaccination. So if we want to slow down transmission, unfortunately, vaccination does not eliminate transmission. So people still need to use those other measures of, physical distancing, masking, limiting gatherings. Definitely we don't want to be saying, oh, I'm vaccinated, so I'm fine, I don't need to do any of those things. That's not the case. You can still get infected and you can still transmit. Luckily, it is protective against severe disease. But I think we've already, for example, the senator, unfortunately, who passed away recently, she was vaccinated, but she had an underlying condition in her lungs and unfortunately succumbed to the virus. And there's a chance that, she may have acquired the virus from someone who is, we just don't know, right? It's possible the virus can be transmitted between people who are vaccinated. And if someone is at risk of a severe outcome, it can have a significant impact. So we still need to bring in all those other measures. So even if transmission does slow down, and even if the rate gets below zero, we still can't become complacent. And I think probably a lot of people are aware that the term that's being used now is re-entering the fourth wave. So numbers are starting to go back up again, and they will fluctuate. So I think we need to bear that in mind also as the holidays approach, right? That we can't just say, okay, I'm vaccinated, so I don't have to worry. Delta has really been a significant setback because it is so incredibly transmissible. And I'll put in another plug for vaccination, global vaccination, because we will keep chasing our tail, going round and round, reboosting. And until the rest of the world is vaccinated, more VOCs will keep coming up and continue to spill over into vaccinated populations who will then transmit it amongst ourselves. So even if altruism should be the main goal and purpose of vaccinating everybody, but secondarily, it's also a means to protect our own population as well.
Thanks, Sam. Just picking up on you mentioned possible severe outcomes of COVID. And, I don't think this is new to COVID, right? This is something that happens with other viruses. That's why we promote influenza vaccination from year to year. While, something can be, fine for someone who is young, healthy, or just healthy, it can have some severe consequences, even if it's the flu virus. And so I think the key message around promoting vaccination in general, just getting our herd immunity up and promoting vaccination is important for COVID. And again, we'll plug the vaccine for flu is out there right now too. And we definitely do have influenza circulating, although, not what we're used to and at the levels, but it still is out there, and getting vaccinated for all vaccine preventable diseases is of very much importance for our population and the global population.
I think one other aspect to consider too, even if it's not thinking about severe disease in the immediate term, in the acute term, think about what we're calling post-COVID conditions, or some people call it long COVID, right? That's going to have, it's such a substantial burden, both for the individual, but also, healthcare systems and society in general, right? So if vaccination can also prevent long COVID, or in some cases, I know there's been some discussion about it even improving long COVID, that's another really good reason. So trying to it won't eliminate transmission, but it could reduce it. It could reduce acute poor outcomes, and it could also reduce the development of long COVID.
Sam, maybe you can just touch quickly long COVID, describing a little bit of what that looks like, just for those that may not have heard of the terminology before.
Yeah, I think people are starting to understand the heterogeneity of it as well, right? So people are using the term maybe post-COVID conditions. And I know, Rob, you've also thought about long COVID as well, so feel free to jump in. But I think that there can be a range of conditions. There's certainly individuals who take a long time to recover, but who do ultimately recover. Like they might take a long time, being more than 3 months, for example, but they do ultimately go back to their previous state of health. And then there are other individuals who've had significant sort of quotidian or daily impact in terms of, different reports, but fatigue, brain fog, many symptoms that have just kept them from going back to their normal level of activity. Rob, you may have something to add there.
No, I think, Sam, you covered it really well. It's, we still don't know a lot about it, and we need a really good definition. We need to keep studying it, but I don't think anyone would argue that it is a condition that we still, as of yet, not fully characterized, but people are definitely suffering from it, and it does not seem pleasant at all.
Thank you for that, both of you. So Rob, maybe I'll come over to you again. So, is there a difference between boosters and multiple doses of vaccine?
Well, yeah, I think this is like one of these things, like, 6 of one, half a dozen of the other. If you start getting a vaccine multiple times, I guess you would really just consider that a booster. I think that if people are sort of wondering, well, if you think about, like, some vaccines we get multiple shots of, for example, MMR, we get multiple shots, and then that's, multiple doses, and that's you're good. Whereas other ones like flu, we need a dose every year, which I guess you could kind of think of that as almost like a booster. So I don't know if there's a really set way of differentiating the two. So I don't know if that's helpful or not.
No, it certainly is. I think we keep hearing the terms used interchangeably and so I can see how people would get confused when they hear it from one source as boosters and others as your third dose or your multiple dose. So I think that's important. So thanks for that information. Building on that, during your presentation, you also spoke about individuals who may have had mixed vaccination series, and now they're maybe going to get their booster or that next vaccine. Again, do you want to comment on, does it matter which vaccine they get? And, do they have to wait 6 months after a second dose to get that third or that booster shot? What is your thoughts on that?
Well, I can only comment on kind of what the science says. And there's, I would encourage everybody to, there's lots of good sources and lots of good data. I know that NACI and Public Health Ontario both have excellent data that, easily accessible reports that it's, you can just Google it and be able to find it. So yeah, I think I'll kind of defer to them on sort of, that thing, but rather because, these are people who they're not humble microbiologists. They are experts on this, who spend their days thinking about all the potential iterations.
Okay, thank you. So the NACI that you're referring to, in case people are interested in searching that themselves, that's the National Advisory Committee on Immunization, NACI. And that's what Rob is referring to in terms of follow-up for more specific guidance with respect to when to get your next shots. And again, sometimes this information is changing. What you hear one day, there may be some direction or more clarity coming, or anyway, that's the source. That's the gold standard, or the source where the information is going to be accurate.
I will say that, again, from the scientific papers that I've come across, that there is great data showing that if you get a third dose of the same vaccine, that it works very well. And there's data showing that if you mix and match, that it may also provide it may provide better long-term immunity. We won't know until we do these studies a little more with a little more longevity, but there's nothing that's published that says, well, if you got 2 Pfizer and then you get a Moderna, they cancel out. Nothing like that whatsoever. So I'm actually due for a booster soon, and I'm scheduled, and whatever they put in my arm, I'm happy to take. In fact, I kind of hope it's one of the other ones. I'll do an N of 1 experiment on myself to see how well it works.
Thanks for putting yourself up in science for us there, Rob. So I'll stick with you on this question. This has come up, and I hear this, and I certainly feel like this is the case. The question is, will COVID-19 one day become a seasonal issue? It feels like it's here to stay. Will it be, instead of a pandemic, will it transform and start to just be endemic? Rob?
If you'd asked Rob from April 2020, he would have probably said, yeah, we won't, we'll eliminate this thing. But, Rob of November 2021 is a little more sober in his opinion. And unfortunately, I think it is something that we're stuck with. We look at some of the seasonal coronaviruses, the ones that cause, the common cold, and they were all zoonotic pathogens. They made the jump into humans. They may have had bad seasons when they first jumped over, and they've stuck around. So there are immunologists who are saying maybe one day COVID will evolve to that. So the next generation, it will, they'll get vaccinated and it'll just be like a mild cold. And I think that's what we all hope for. But there's actually a really good, I would encourage people to read. There's a really good article by Ed Young, who's just a top-notch scientific writer in The Atlantic, who kind of deals with this and addresses this. And he says it may be kind of like what we see with flu, where some years it's a really bad season of COVID, at which point people will have, more masking effect, or people will choose to socially distance, and everyone will make sure to get their boosters. And some years it will be, less of a risk, or it may depend on your age. So I would encourage you to read that. He really actually talks to a lot of very smart people who are thinking about this, and summarizes much more eloquently than how I just said it.
So Rob, there's some hope for the future where we might see a bit of a normal state where it becomes endemic, and hopefully more like the other coronaviruses that circulate. But again, more time will take more time to get there and understand how this will evolve. So thanks, Rob, for that.
I see Sam wanted to throw something in.
Oh, I'm so sorry. Go ahead, Sam.
I was just going to quickly throw something in. I could take no credit for. It was actually a colleague who pointed this out, that even if it does become endemic, if we think back, if it's possible, to pre-COVID times, how bad flu was, the annual crunch of, hallway medicine, how overwhelmed the system could get even in a bad flu season. If we layer SARS coronavirus 2 on top of that, we'll probably be it'll feel like an ongoing pandemic, right? And I think that kind of goes back to the need to just reduce as much as possible the net burden of respiratory virus infection, flu, RSV, COVID-19, all of the above. So as individuals, we can get vaccinated with the vaccines that are available. And as researchers and as funders, we can try and promote, development of universal vaccine for influenza, for example, development of vaccine for RSV. There's a history there that's been fraught. It's a challenging history, but people are willing to dust off the lab books and look at it again. And I think that's pretty critical. So we can avoid those annual crunches of hallway medicine who knows with COVID, but with flu, it was December, January, February, where things would really get messy. And we would all feel it system at the system level during a bad flu year.
Thank you, Sam. So Sam, maybe this question's for you around how do we identify the variants of a specific virus? And based on what we know right now, do our vaccines sufficiently protect us against the variants that are currently circulating in Ontario or nationally in Canada?
Yeah, so there are a couple of ways we can identify the variants. Rob was very involved in all of this as well. You can, at first, when there were a limited number of variants of concern, we could look for specific mutations. So we would do PCR screening for specific mutations. For example, the 501 change in the spike protein is one example. But then all these other variants, which may or may not have that change, would come out, and then they also came out in rapid sequence, right? So you would adapt your screening, and then another one would come out, and then you would adapt your screening, and another one would come out. So ultimately, whole genome sequencing, which I showed during the talk, where you kind of sequence the whole virus from one end to the next, will show you exactly what the story is and everything in it. The key is to do it in a timely manner. If you have weeks to months from the time you receive the sample to releasing the sequence, it's not really very helpful. So a lot of us are working to do that fairly quickly. There have some of the variants are definitely they've definitely escaped some of the monoclonal antibodies. So for example, one that was called bamlanivimab that was developed early on is no longer effective because of the changes in the variants. So that's just a monoclonal antibody. It's not a vaccine. There has also been an impact on vaccine efficacy, and it depends on the variant. So it varies based on the variant. Some of them, there's less of an impact. Others, there's a greater impact, and it really depends on where the changes are. If they're in those antigenic sites that antibodies are raised against, then there's more likely to have for there to be limited effectiveness. One thing I don't think we quite know yet, so far we've been repeating vaccinations with the same original spike sequence. We haven't switched it out yet for a more updated one. Part of that is that the virus just, like, which one do you pick? For a while, I mean, we're now at Delta dominating, but obviously we got Alpha, Beta, Gamma, Mu, Delta sublineages, Delta plus. So it's very much a moving target, and I think things might have to settle down before we say, okay, this is the strategy we're going to use. So for influenza virus, the strategy we use for that is we look at what's circulating in the other hemisphere, because the seasons are opposite. And then we use that. So we look at what was circulating in the antipodes, so Australia, South Africa, etc. And we put those viruses into our vaccines. That's usually how it works from year to year. And the WHO does this on a regular basis. I have no idea whether that's going to be the strategy for SARS or not. Different viruses, different molecular clocks. I really don't know. But needless to say, we do have to keep doing the science. Every time a variant escapes, people are looking at how antigenic immunized serum. So people who've had immunization, they take their serum and they look at whether or not it neutralizes the virus, and they do animal experiments and other. So phenotyping the variants, that's what it's called when you look at the variant characteristics at that level, is really important, and it's important to do that very quickly.
And it's great to know that there's people like you and Rob that are out there working on all these pieces of the puzzle, as well as our other colleagues. So thank you for that, Sam. Now, what about Rob? We've got influenza virus circulating in the community. We've obviously got SARS-CoV virus circulating in the community. Can these two get together and mutate and create something new?
Thankfully not. I mean, sounds like the great plot to some sort of sci-fi film. So that's my movie right there. But no, they are just the gene. So not only are they not part of the same viral family, but their genomes are different. Influenza is a segmented genome, so that it actually has 8 pieces. It's a negative sense, whereas SARS-CoV-2 is a positive sense, single strand RNA genome. So it would be the equivalent of trying to put a dog and a cat together and get them to have some sort of hybrid. So safe to say that we don't have to worry about those two sort of making some sort of super virus. That being said, each of them are mutating on their own, and there's certainly the potential for each of them to be pretty nasty.
Thanks, Rob. Yeah, Sam, this one might be, I think I saw it in your presentation, the viral genomics and the trees and a little bit of sort of, when I think about how we investigate outbreaks, we're looking to see if we can figure out who the source is and doing some sort of genomic typing to determine that. Can you explain how that helps with contact tracing, that information, or how it's done?
Yeah, I mean, ideally it would be done very quickly so you could do the contact tracing, but that's not always possible. One of the key applications would be if you have a large outbreak and you're wondering, well, are there just multiple introductions, or is it actually passing from one person to the other? Because the interventions might be different. And Natasha, you would know more about that than any of us. It really depends. If there's enough variation in the genome, you might be able to see these, what we call single nucleotide polymorphisms. Some of them can be low frequency, and you can show relatedness of the virus or lack thereof. It's almost more helpful if they're unrelated. You can say, okay, these ones aren't related. It's when they look really closely related, but it's a common variant that is just ubiquitous, right? It becomes a little bit more challenging. Now, you can optimize it by sequencing what we call deeper and looking for more of these low frequency variants, but it's all still somewhat inferred. You can't say 100% for sure. There was a period of time where there was a lot of heterogeneity in the viruses that were circulating because there were so many introductions from so many different places that they were really clearly unrelated. Now everything's Delta. Though we're starting to see some sublineages. So it's certainly useful, but there are limitations, I think, is how I would word it. You can't say definitively, if you're wondering if for example, there's a daycare outbreak and you're trying to pinpoint the source, it always has to be compared to baseline data from the community, which is why always doing sort of a baseline amount of sequencing is important. Rob, you've been doing a lot of sequencing too. If there's anything you want to add there.
No, I think you really covered it, Sam. It's one more tool, but it obviously has to be paired with the experts in infection control. And I think I do really enjoy the collaborations that we can do, and we all put our heads together and help with our little piece of the puzzle. And so it's impressive that we have these tools available now. They don't give us all the answers, but it is kind of nice that they do give us some.
Thanks, Rob. I'm going to jump on the tools because honestly, I think a lot of times when we're managing outbreaks or trying to do any sort of contact tracing, sequencing and this type of work really does add to our tool belt. It doesn't necessarily, we don't hang our hats on it, but certainly it helps to try and explain transmission. Sometimes it helps to understand the dynamics of an outbreak. And sometimes that even things though that are historically looked at, we can use some of what we learn from that to apply it to future situations. So I think definitely from an infection control perspective, we find these elements helpful, but it is somewhat challenging to get them at the moment in time when you need them at that, in place to inform decision making. So sometimes we add it to the layers, so to speak. Thank you for that. So Sam, maybe you can talk a little bit about some of the developments you made since isolating SARS-CoV-2, Rob as well, since you both collectively worked on that. What was the experience like?
It was wow. It's hard to describe that experience. It was like nothing else. I mean, one thing that really struck me at the beginning was that when you think about it, I could probably count on one hand the number of people in Canada who had worked with high consequence coronaviruses in biocontainment. And I think that really shows what a dearth of capacity we had when this started. In some ways it's understandable because there are a limited number of facilities you could do this work in. And there are obviously some very important regulatory issues. But when you have so few people who can work on specific pathogens, that almost becomes a public safety issue, right? And it still exists now. Like if you were to ask me, are we ready for an H5N1 outbreak or pandemic? I'd say no. There's no one in Toronto who can work with H5 at the moment. Our facility can't take an H5 virus. It's what we call a security sensitive biological agent, and there's nobody with that clearance to do that work. And there are probably only a couple places in the country that can do that work. So we were very lucky that the stars aligned and it happened, but it really could have been a very different story. That was an exception and not the rule. So just a plug to, whatever capacity has been built now, to keep it and to continue expanding on it. And then since then we've worked closely, and great thanks to Alison McGeer, who's been sending us samples from patients to continue isolating virus. So we've continued to do virus isolations focusing on the VOCs, and we're looking at doing transmission, or we are doing transmission experiments to look at why some of these viruses transmit better than others. So those have been some of the key activities. And then the other key activities, I didn't talk about the data per se, but we've been doing a lot of wildlife surveillance with colleagues at the Ministry of Natural Resources and the Canadian Wildlife Health Cooperative. So we've kind of gone from bedside to lab, but then into the field and back to the lab. So that's sort of what we've been up to, and along the way helped out with a few things. We're not, myself, I'm not leading any antiviral projects, but we're helping a few people here and there. I know Rob's doing a lot along those lines and with vaccines too. So Rob.
Yeah, well, I'll say that I think that Sam's right that it was quite of a stroke of luck, but I think it was I was quite lucky to work with great people, Sam and our colleague Aaron J. Banerjee out at Vito, and other people. So many people helped out, and I think that what's that Pasteur quote? Chance favors the prepared mind. So I think we were lucky, but yeah, we had a good team, which increased our chance of things working out. And since then, I feel like I've lost track of all the different projects, everything from helping out with point of care testing. I've been doing some, as Sam alluded to, a little bit of some antiviral stuff and some monoclonal antibody work with some colleagues. Both of us actually were lucky enough to work with a company that's developing a Canadian mRNA vaccine, which is, I think, super cool. I'd love to see a Canadian vaccine out there. So yeah, I could spend 2 hours, but I won't. So it has been a whirlwind.
Thanks so much. I'm just looking at the time, and I'm going to try and keep it. We've received so many amazing questions and questions that we hear quite often. So this is great engagement from the public here. And I just, wanted to maybe pick on a couple of them that jump out at me as being questions I hear very often, just to bring us closer to the end of this presentation and question and answer period. So maybe I'll jump to Rob, how is the vaccination schedule determined? And is there any concern about too much time passing between doses? Do you have to restart your series or just continue on? What's your opinion on that?
So it's a great question. Again, this is the kind of stuff that usually takes people like to develop a vaccine schedule. Usually takes many experts, many years, working away at it. And again, there's great literature from NACI and Public Health Ontario. So and there's more than one factor, but like you often have to calculate things like when's the optimal time to not just prevent mortality, but impact on people's lives, that kind of thing, cost, all that is factored in for determining it. And unfortunately, I wish I can't give it how, if and when a schedule will be done for COVID-19, and even the idea of restarting your schedule. We probably won't need that for COVID-19 because likely it'll be like flu. We'll get it every year. For other vaccines, it's really vaccine specific. Hepatitis B, they do recommend restarting your dosing if you are a non-responder, but something like Tdap, they don't. So it's very specific to the vaccine, and unfortunately, I can't give an all-encompassing answer.
So NACI, follow NACI.
Follow NACI.
Again.
That's right.
So, on that point, Sam, maybe you can elaborate. Lots of information. I think, Rob, you said it best. Don't follow the Twitter. What did you call them again? The
Twitter trolls.
Twitter trolls. I for sure, I agree. Sometimes I become overwhelmed with the posts that I see and just whether to engage with them or not. But regardless, I think, where can we get really good, credible information about vaccine development, Sam?
So there are a couple of great sources. I think Rob already mentioned Public Health Ontario. Lots of really good synopses, and the Ontario Science Table has also put out really good briefs about really a wide range of things if interested. And those tend to get updated quite a bit. Public Health Agency of Canada, the CDC in the US. If for some of the more granular stuff, if interested, Can COVID is a good resource. And then also there's the CITF. So that's the Canadian Immunity Task Force. You might see some more specific papers and some of it's quite detailed, but a lot of the researchers who are working on vaccines and immunity are part of that task force or are funded by that task force. So it's a pretty good site to go to if you're wondering, okay, what's going on right now. At a more global level, there are other sources. For example, the University of Minnesota CIDRAP. I can't remember what CIDRAP, CIDRAP stands for, but they'll often post if there's a really important press release or preprint or a paper. You can find it on their website. I used to follow the preprint, both Med and bioarchives, and there's so many now that you need someone to distill it. I actually find the Nature briefs really helpful. So Nature and Science. So obviously the articles themselves can be really dense, but both Nature and Science put out really nice summaries, like lay summaries of the science. So if you're looking for like an overview or an update, those are both really good sources as well.
Great, thanks so much, Sam. And I'm going to bring us to the last question here. And then I've really taken a couple different questions and I'm melting them together. And I think it's really all encompassing of both presentations as well as just sort of that global perspective. And, to that point, where are we with emergency preparedness? I'm going to look to both Sam and Rob about this question as well. I'll add a little bit of my experience, but really, where are we at with respect to emergency preparedness, preparing for future public health issues when it comes to the vaccine, when it comes to just preparing for the next pandemic in general? Is there anything that we can do to stop pandemics? Like that sort of thing. And I think, when I'll start and then I'll head it over. But some of my wonderful experiences early in my career included working in public health where I did work at a SARS assessment clinic back in 2003. Just what really struck me is the importance of strong collaboration on multiple levels. We're talking about global levels here, but we're also talking about community levels. And really, that really pushed us and drove us into pandemic preparedness and really inspired a lot of great conversations between healthcare facilities, community health, Ministry of Health, so on and so forth. It really did start to engage a lot of people in conversation conversations about where we would be if we had a pandemic and how can we collaborate and plan for a pandemic. At the time, we thought it might be an influenza pandemic, but needless to say, all that preparation work was really, a phenomenal backbone in how we've been responding to the COVID pandemic, and really had always kept those wheels in motion, even though we were anticipating, and hoping not to have a pandemic for sure, but always on edge and making sure that we had the right surveillance in place and enhancing surveillance. There's so many conversations that we can have here just about general preparedness and emergency preparedness. We can have a whole presentation just on that alone as well. But maybe Sam and Rob, I'll punt it over to you for your last thoughts, and then I'll wrap it up for us today.
Well, I mean, you could look at it from different perspectives, but if we start at the most microscopic, which is the virus, I think in terms of surveillance, I think that that's something key. And if we do surveillance, we're going to find things, right? So having the tools that are available to us to figure out, well, what's important and what's relevant, what's the risk associated with whatever it is that we find. So good old-fashioned virology, biology, we can't forget those. And I think that it's really critical that we coordinate those efforts a little bit better. It's not, unfortunately, like the containment level three labs across the country, got together and coordinated an effort to try and address this pandemic. And in a way, in a country like Canada, there's really no reason why that can't happen. So I think that we probably underutilized some of our resources to a degree. So I think that could be improved significantly. And then on the other side of things, still sort of sticking with the microscopic, is around biomanufacturing. We really relied on other countries for our vaccines. We really rely on other countries for antivirals. If you look at the AbCellera story, they actually licensed their intellectual property to the US, and now we're buying back Canadian IP, or we're buying back Canadian IP that, had we had the manufacturing capacity in Canada, probably just could have stayed in Canada and maybe helped the rest of the world. So I think that that's another issue. Then if we take a step back, like macroscopically, and think back to what some of the drivers of spillover are and how to prevent the next pandemic, essentially what we did was we established the ideal conditions for a pandemic, right? There was a spillover that, almost no doubt was enabled by human behavior. And then because of our rapid global travel, we very effectively spread that virus all over the world really quickly. This is human, like this is us doing this, right? So we need to rethink normal, because what was normal is what got us here. So we need to think about, how do we change normal so that we're not back here in another however many years? And just think about some of the fundamental drivers that sort of I talked a little bit about. In fact, someone raised this, and I thought, oh God, no, I don't think it's likely to happen, but someone raised the risk of associating the risk of flooding with avian influenza. And I was like, what's happened that's happened in other jurisdictions, not in temperate regions, but more tropical regions. So may not be a risk, but if we look at extreme weather events and the impact on zoonotic diseases, there are some pretty clear, tight links there, right? So we need to address climate and other things. So I know that's really big picture stuff, but we need that kind of paradigm shift, because again, if we keep going the way we're going, we'll just be in a spiral of major events, be it zoonotic or atmospheric or meteorological or climate.
For sure. I think adapting to lessons learned from this pandemic and previous experiences with the infectious disease outbreaks and pandemics is certainly, I think, valuable information, and with that whole global perspective that you're talking about as well with One Health. So thank you for that, Sam. Rob, anything to add?
Well, I guess maybe just three things. I think Sam made excellent points, many of which would definitely be on my list as well. And I agree with you too, Natasha, that throughout the pandemic, we faced a lot of challenges, and we had to, in the lab, we had to rapidly figure out what was working and what wasn't, and change and adapt. And there wasn't time to say, but that idea worked yesterday. Why is it? You just had to move on. So at some point we will have to do that with this one, and sort of check our egos and say, what did we really do right and what did we really do wrong, so that we can fix this. And I would tell people that I would encourage people and members of the public to remember to take this threat seriously. And there's, as Sam alluded to, there's no reason that we wouldn't see an avian influenza pandemic tomorrow. There's nothing in place that's going to guarantee us. Viruses don't care that we've just had a pandemic, or that it's an election year, or that we all have plans for Christmas. So, and this is something where the public, I would encourage you to hold our leaders accountable for this. This has shown us that a virus was able to undo the fabric of the world, essentially. So it's something that we should always be asking, what can we be doing better, and what should we be doing? And I think my last point will just be that we need to support scientific research, not just here in Canada, all over the world, and not just in all aspects, everything, not just virology, all of it, because what we saw from the mRNA vaccines is that it was 40 years of research that got us there. That, some of the new antivirals that are coming out are things that people started a decade ago, and, plugged, and had they, not persevered and said, not found a way to keep it going, we wouldn't have it now. So we need to sometimes support that, just in the hopes that it will be useful in the future. And again, that's where I think we need the help from people in the public to pressure the people to hold the purse strings and say, like, hey, are you making sure of it to make this a priority?
Thanks so much, both of you. We've now come to a conclusion of a very fruitful conversation, presentations, and conversations related to the questions that were provided by the public. Thank you again to everyone who was able to send in questions, and to the speakers for facilitating such an informative discussion. So, to our audience members, we hope that tonight's session was educational and meaningful, and relevant to our current circumstances for you. Please be sure to take a moment to fill out the electronic evaluation form that's at the end of tonight's event. This will help us plan for future topics and that are of interest to you. And I would again like to thank our speakers for an incredibly instructive evening, and a big thank you to all of you for joining us today.